Hi, thank you for tuning in. I'm Robert Aboukhalil, and today I wanted to present sandbox.bio. This is a web application that features interactive bioinformatics tutorials. And specifically, I wanted to touch on how we've been able to leverage new web technologies to enhance bioinformatics education. So as we all know, tutorials are really important for bioinformatics education, and whether they're text, video, or in person, they sometimes have similar challenges. One big challenge that we have is setting up an environment. So if you've run bioinformatics workshops before, you know how challenging this can be. Bioinformatics tools are notoriously difficult to install, whether you have Conda or Docker or notebooks. Everyone's environment is always a little different from yours. You know, some students have Windows, others have Macs, some have M1s, some have a few of the dependencies pre-installed, but not all of them. Others have, previously installed Conda accidentally and can't exit out of their environment. So setting up an environment is time-consuming, it's difficult. And there's also sometimes limited interactivity. Right, you generally can't directly interact with the tutorial. You have to go back and forth between the teaching material, copy pasting the commands into your own environment, trying to replicate what you see. And speaking of copy pasting, it can be scary to make mistakes because you are running these commands on your own system. And so, for example, if you had a tutorial where you had to define the name of a directory inside a variable, which it then tried to delete, you can imagine how this would not end up being very good. Now, of course, this is a made-up scenario, but the fear there is definitely real. And to me, it's kind of an important part of learning. Like when I'm learning a new bioinformatics tool, the way I really learn is not by following exactly what the tutorial says, but at certain points, deviating from what the tutorial says and tweaking the parameters, seeing why they matter and what they do. And so this is where sandbox.bio comes in, and it tries to address some of these challenges. And so the idea with sandbox.bio is to have a web interface that brings together the command line that users are used to and the tutorial content. Bring those two in one place where the dev environment is already set up, ready to be used, all the tools are installed, and all you have to do is read the tutorial and click around. And so, for example, with permission from the Quinlan lab, we took this really good BetTools tutorial that was in text-based format and adapted it into sandbox.bio such that it looks something like this, where on the left you have the original tutorial contents, and on the right you have a command line interface in which you can play around with, you can type commands, you can execute them. And so as you're following this tutorial, what you can do is click on these boxes that have commands in them, and it will then execute them for you and show you the result. And now, this is not a simulation, it's actually running BetTools directly in the user's browser. And so you could see that if, for example, I go into this command, modify it a little bit, add a -v. So instead of asking BetTools for the intersect, I'm asking for the opposite. Give me, within these two bet files, give me regions that do not intersect, and I press enter and it will execute that for me and give me the answer. So, I can deviate from the tutorial, try things out, test new parameters, and see what it does. And just to, show you that this is the real BetTools running there, you can type BetTools version, you can type, any other command that usually works outside of the command line. And so at this point, what we have is definitely an interactive interface. The tutorial and the CLI are both in a single place, and you can interact with them, play around with it. There's also no environment to set up, right? You just go to a website. As soon as the page is loaded, you have everything that you need in order to learn. And finally, you can experiment as much as you want to, because this is a sandbox environment, as the name would suggest, that's running inside of your browser. It's not touching your actual files. You can run RM as much as you want. It's fine. You just refresh the page and it'll reset for you. And one of the neat things is that we can now extend this to not just include tutorials where you follow steps, but also exercises where you apply what you learn in order to see whether you kind of got the concept. And so here I'm showing the example, the tools exercise, where what we're asking the user to do is use what they've learned to solve a particular problem. And so what this now lets us do is show the user a few steps for what they actually need, the criteria that they need to fulfill in order to complete this exercise. And once they do, for example, fulfill one of the requirements, they will be able to see which ones they have to fill, and they can keep going until they get the right answer. So this is kind of a neat way to guide the students along the way and try to help them learn on real life exercises. So how does this actually work? Well, what we do is we take C and C++ genomics tools, like that tools, SAM tools, Bowtie, all those, and we can compile those tools from C++ to WebAssembly. And WebAssembly is what lets us run these tools directly in the browser. And so we actually use this library called bioasm. So this is an open source repository of genomics tools that have been pre-compiled to WebAssembly. So this is a project that I maintain. You can check it out at bioasm.com or on GitHub. And so the way it works is we have all these tools that are in C and C++, whether they're bioinformatics, like vet tools, Bowtie, SAM tools, BCF tools, or general command line tools that we also happen to use in bioinformatics, like awk or JQ for JSON parsing. And so what bioasm does is it figures out a way to compile those to WebAssembly, generates this .wasm binary that you can then use to load these tools in the browser. But what bioasm also does is that it hosts these WebAssembly binaries for you and provides this other tool called Aioli that helps you leverage all the bioasm CDN and run these bioinformatics tools in a separate thread in order to make sure your UI stays performant. And so, just to show you how simple this is a few lines of code here where I'm loading the Aioli library from CDN, initializing it and saying, I would like samtools command, and then you can write a samtools command and it will execute it for you and return the result. And so, here I'm showing a simple example, but Aioli lets you specify multiple commands. You can load as many bioinformatics tools as you want. You can also mount local user files. So if users give you a local file, you can mount it on a virtual file system, which will simulate a file system, but within the browser in memory. And it also lets you mount URLs as well. So yeah, besides sandbox.bio, there are other tools that use bioasm in order to run bioinformatics commands inside their apps. And these are not even bioinformatics education tools. They're just general bioinformatics web apps. So in any case, please check it out if you're building web applications and you think you could reuse some command line tools in bioinformatics. And if you have any questions, feel free to reach out. More than happy to collaborate on that. And so going back to sandbox.bio, now that we use bioasm to get these WebAssembly modules, when these modules are downloaded onto the user's computer, that's kind of the end of the communication between the server, right, and the computer, because at this point, any BetTools command that you run executes locally in the browser through WebAssembly. And so if you run a BetTools intersect command, it will give you back the answer. And the web app does not have to contact the server in order to run that command. Alternatively, a more traditional architecture would have looked maybe something a little different, where the web app would make an API request to our server saying, I would like to run this BetTools command. The server would then have to find some available worker nodes, give them the command, and execute it, and then return the result to the user. So as you can imagine, this can get a bit involved. And so the reason why sandbox.bio uses WebAssembly is primarily because we can run all the computation on the user's computer and not have to maintain our own servers. And so the advantages in our case, this is much, much cheaper and much more scalable. Using WebAssembly, you can very easily scale this to thousands or tens of thousands of students, even more, because all you're doing is providing each user with static WebAssembly assets. These are just static files hosted on a CDN. Very easy to scale, very cheap to host, compared to having a cloud architecture that has to scale up and down, execute arbitrary commands. That's where the security comes in. It's a little harder to make sure that no one's minting NFTs on our servers. And so we don't have to worry about that, right? Because everything runs on the user's computer. And in most cases, this means that the tutorials are a lot more responsive, right? Because you type a command, you press enter, it executes right there. It doesn't have to go make a network request and then wait to get the answer back. Now you may be wondering, okay, this is all great, but what are the limitations of this approach? Well, there aren't. I'm just kidding. Of course, there are many limitations. So first, because you're running everything in the browser and every user's computer is different, you can't assume that everyone has the capacity to analyze a lot of data. And so one limitation is you can't have tutorials that need a lot of data. And so, this in practice is not so much of a problem because you can always downsample the data. And then you have a tutorial that's still conveys the relevant information and knowledge, but just runs a lot faster on most people's computer. One limitation that is a lot more real is, some tools need a lot of RAM, right? And WebAssembly currently is 32 bits. And so you can only use up 4 gigs of RAM. And so there are certain tools that simply need more than that. But even if you only need 2 gigs of RAM, that's still quite a bit of data that you're loading into the user's memory. And that's probably not a great user experience. So for now, we don't support these kinds of tools. So you can imagine, if you're trying to assemble human genomes or aligning RNA-seq data using tools like STAR that use a lot of RAM. These kind of tools are not yet supported for that reason. Another limitation is that right now we're only porting tools written in C and C++ to WebAssembly. And so tools written in other languages, like say Python, are not yet supported. We can also have pretty good support with tools written in Rust as well, but there are slightly fewer of those than C and C++. But that's certainly one thing that we want to expand to next. So speaking of what's next, we basically would like to add a few more features, and try to address some of the limitations that I just mentioned. And so one thing we'd like to do is to test out moving from text-based tutorials to video tutorials. And so we're collaborating with the Genomics YouTube channel. So this is a popular bioinformatics channel on YouTube, and it would be interesting to integrate little bits of videos across a tutorial to kind of make it more engaging. Some people prefer video over text, and so that's something that we're trying out. Another thing we want to do is allow users to select local files and be able to interact with them in the sandbox. So you can imagine, say you just completed a Bet Tools tutorial, and you learned about all these new tools, and you have Bet files on your computer. The obvious next step is, how do I apply what I learned to my own data? And so it would be nice if the user could essentially provide local files to sandbox.bio, and then they could test out running Bet Tools commands on those files. Now, of course, there are some limitations as to, file size, but we can always get around that by providing only a subset of the file. So that's something that we're looking at. Another thing we really want to do is enable tutorials for Python-based tools. So there's a project called Pyodot. And basically what they've done is they've taken the Python interpreter, which is written in C, and they've compiled that to WebAssembly so that they're able to run Python code in the browser, which is fantastic. And so we want to find a way to integrate this project into sandbox.bio such that we can have tutorials that are not just about C and C++ tools, but also Python-based tools. Now, the last thing, this one's a little more tricky, but one big limitation of sandbox.bio is that you cannot have tutorials for tools that are written in languages that cannot be compiled to WebAssembly. So for example, if you have a pipeline that uses Bash and then Perl and then R, that's just not going to work. And so one thing that would be interesting is to look for a cost-effective way, importantly, to have some of these pipeline executions be done in the cloud, but try to do most of it still in the browser for the parts of the pipeline that can be compiled to WebAssembly. So that's more long-term. It's a bit more involved, again, because cloud architectures can get very expensive very quickly. So there has to be some really intelligent tiering, so to speak, of which computations do you do in the cloud and which can you do in the browser. So that's all I had for today. Please check out sandbox.bio if you're interested in collaborating, whether it's adding new tutorials or converting your own tutorials into sandbox.bio. Please reach out. Also check out my other project, BioAsm. So if you're building web applications and you can make use of bioinformatics tools, you could check that out as well. And with that, I want to thank you very much for taking the time.